TCR gene editing is an approach that was proposed for the first time 10 years ago. And we propose it with a project performed between my group and the group of Luigi Naldini in San Raffaele. At the time, we collaborated with Sangamo, and the tool was zinc finger nucleases. Today, CRISPR-Cas has made the TCR gene editing much easier, much more efficient also. The concept is anyway the same, independently on the tool that it is utilized. And it is it's an approach by which we do not only add a new specificity to T cells harvested from a patient, but we completely redirect the specificity of a T cell. So let's do a step back. How can we make a T cell able to recognize a cancer cell? We can use mainly two approaches. One is to use a CAR, a chimeric antigen receptor. These are really successful already in the market. And whenever it is possible, we use CAR T cells. But unfortunately, CARs recognize only antigen present on the surface of cancer cells. We cannot really see a target that is inside the cancer cells with the CAR. And many targets in cancer cells are inside the cell. So if we want to target a molecule that is inside the cancer cell, we need to use a different approach. TCR is a good alternative because a TCR recognizes a little peptide presented by MHC molecules. So even intracellular molecules are transformed, are processed in these little peptides and then presented. So TCR is a good alternative, but when we add a new TCR to a T cell, we face the problem that every T cell that we manipulate originally, before the manipulation, has its own T cell receptor. And so we end up with a product that has 2 T cell receptors, potentially even 4 because of mispairing. So we don't have control, not of the level of expression, the efficacy of our cells, and not even of the safety profile, because mispairing can lead to autoreactivity. So TCR gene editing is an approach by which we do not only add the tumor-specific TCR, but we really substitute the specificity. We delete the endogenous and introduce the new one, having a much more controlled T cell products.